Here we are at Chapter 8, Bio 112 with Ms. Hook. This chapter we're going to look at mitosis and meiosis, the different ways cells can divide and send their DNA on. Your book likes to relate things that we're learning to things that are going on in society. Here's a great example. So, zookeepers at a Chester Zoo noticed that their Komodo dragon, just a single female, had laid eggs. And those eggs, they developed. So even though they hadn't been fertilized by a male, they were viable offspring with only her DNA. And that process without being fertilized by a male is called parthenogenesis. So let's look at cell reproduction and why. Okay, so cellular reproduction, it can be the birth of new organisms, but most commonly, like especially in our bodies, for example, it's generally just going to be the creation of new cells. And so what happens when a cell goes through reproduction is you make what are called two identical cells that are two daughter cells. They're exactly identical to the original parent cell and each other. They've got the same organelles, the same DNA. Everything is exactly the same. So how do they do that? Well, to get DNA to both sides, the exact same copies, you have to go through a very specific process where you multiply the DNA by 2 and then divide a copy to each new cell. And how they do that is they duplicate their chromosomes. Chromosomes are their DNA structures that are these like thick, condensed versions of the genes that make it easier to separate them to each new cell. So these two cells originated from a parent cell. They took their chromosomes, made copies of each, and they sent one copy of each of the chromosomes to the new cells, so that once they're done dividing, they look like the original parent cell too. So when would we want to do cell division? We'd want to replace any damaged or lost cells. So if you cut your skin, you'd want to replace those cells there. You'd do cell division to grow larger and for reproduction. So generally, cell replacement would occur like in adults. Anytime you wanted to replace your like kidney cells or your skin cells or your liver cells. And then growth would occur during development. There are two ways to reproduce. There's asexual reproduction and sexual reproduction, and both have their advantages. So in asexual reproduction, you only need one individual. It's making exact copies of itself, growing new individuals. So for example, this is a hydra, and it has these little buds that come off of it, and those will be new hydra that will be released. Apples are another great example. When you go to the grocery store, there are different species of apples that you can get, different types. Those all came from the same original parents. So if you get a Fuji apple, for example, those would come from the same Fuji plant. And what they would do from the original parent plant is they would take clippings, cut off portions of it, and grow it with the roots of another plant. And then you'd have these exact copies of the original plant. So you'd know exactly what the apples are going to be like. There's no sexual reproduction combining those traits with other apples. Let's look at some more examples. So when you have individual single cellular organisms and they divide, that's reproduction. The regeneration of a sea star. So here there was an original limb, and now it's regenerated the rest of the body into a new sea star. And here's the example of clipping, so putting down a leaf and growing a whole plant from it. So when you do asexual reproduction, the parent and its offspring have identical genes. And this is going to be really important. So if the environment is stable, the parent is doing really well, they would be better off if they could reproducing asexually. That way, if they're doing well in the environment and it's not changing, their offspring should be really well off as well. So what they would do is they would go through what's called mitosis, normal cell division. So it's going to be asexual reproduction, where you're not mixing any genes, and then just the growth and cell division within organisms. Asexual reproduction, on the other hand, that's a lot more complicated. So there you have fertilization occurring. You're taking genes from one parent, from an egg, and the other parent through sperm, and going through a specialized type of cell division called meiosis, where basically the egg and the sperm each have half of the genes. And then when you combine them, you get a whole organism's worth of traits. So if you're a sexually reproducing organism, you use meiosis, or you'll hear it called meiosis as well, for reproduction, for making these sex cells. And then mitosis will be what you use for your normal cell growth and maintenance. So whenever you're dividing kidney cells or skin cells. Egg, sperm of human. You've got a sponge and it's doing sexual reproduction, and there's a frog fertilizing eggs. Let's look at mitosis first. So mitosis is that asexual reproduction and normal cell division within our body for what are called our somatic cells or normal body cells. Your cell goes through a cycle. Depending on where it's at, the cycle can be longer or shorter, where they go through mitosis. So on our human eukaryotic cells, those genes are going to be in chromosomes in the cell nucleus. Remember, you will have in plants chloroplasts and mitochondria. Mitochondria are also in animals. They've got their own DNA because they've got bacterial DNA. So they have to go through division as well, but we're going to focus on that nucleus. So each chromosome, there's a single chromosome, is made up of DNA. And when it's in the chromosome form, it's really compact. You've got these proteins that the DNA wraps around, and so it's all pulled together like a ball of yarn. And on that ball of yarn of DNA, you've got lots of different genes coding for different proteins. So here's an example of the human genome and our chromosomes. So what we did was we labeled them by number. The longest first, so 1, 2, 3, 4, 5, 6, 7 through 22, and then the 23rd set we have is the sex chromosomes. And that chromosome number is going to be dependent on species. And you'll see we've got pairs, right? So for each chromosome that you have, chromosome number 1, for example, one of those copies came from your mom and one from your dad. So each of your normal body cells has 2 copies of each chromosome. Here's examples of different species and the number of chromosomes they have. So you can see that the number of chromosomes is not generally indicative of complexity. The rat, for example, the red Vishkasha rat, that has 102 chromosomes, where people we fit in at 46. Remember we had 23 pairs, so 23 times 2 is 46. Some fern species have thousands of chromosomes because they can reproduce in a way where they can have more and more genes coming in and still be viable. And that would be like extra copies of each of their chromosomes. So normally DNA isn't visible when the cell is not dividing. So for example here, and you can see a little bit on the other side there, those cells are in the non-dividing phase. And you can see where RNA is being made, those are the dark spots there. And you can see some darkness of the DNA, but it doesn't become chromosomes. It doesn't become thick like you can see here until cell division. And here this is a cell wall, so you know we're looking at a plant. Okay, let's look more at that DNA. So you've got your DNA, the double helix, and then when we want to make it into chromosomes and thicken it, we condense it. And what that means is we wrap the DNA around these proteins, these histone proteins. So you wrap it around, and then you wrap it around itself, and then it gets really thick. So when the cell is not dividing, the DNA is thin and spread out so that we can make proteins. We can make RNA from the DNA. And then when we want to divide, it condenses, gets thicker, becomes chromosomes. So it's like the string again. I love this idea. You've got a string all along when you're reading it, and then when you want to copy it, you want to wind it up into a ball so that it's easier to move. So whenever you're making a new copy of DNA, you take each chromosome and you duplicate it. Those two copies are called sister chromatids. They're exactly identical. So here, this is like chromosome 1. There's mom's copy, there's dad's that you have in each of your cells. And then when you want to divide, you make this sister chromatid. And they're connected at what's called the centromere. That connects them together. So when the cells divide, one copy of the purple is going on to the first cell, the other is going on to the second new cell. Same thing with the other copy of your chromosome 1. One will go to the first cell, the other to the second. So that at the end, you'll have exactly what the original cell had, what the original parent had. Yep, here's a nice showing of how that works. Original parent cell has this chromosome. You double it into identical copies, sister chromosomes attached at the centromere. And then for each new cell, you're going to make sure that each new cell gets one copy of that chromosome. So how does that happen? How do we make it work? Well, you've got what's called the cell cycle. And that's this sequence, this order of events that goes through a very particular sequence so that we make sure we get one copy of each chromosome into the new daughter cells. So the cell cycle has two phases. First, interphase, that's the non-dividing phase. And then next, mitosis, the mitotic phase, where the cell is dividing. So most of this time is interphase. This short box here is the mitotic phase. That's where cell division is actually occurring. So interphase first. You've got a daughter cell. It's going to grow. It's got these three portions. So G1, gap 1, S, G2. So during S, that's the synthesis phase. Synthesis means to make things. So that's where we take our original single copy of the chromosome and double it. So here's interphase. We can't see any of the individual chromosomes. These dark spots are called the nucleoli. That's where you're making ribosomal RNA for ribosomes. And during that interphase, we made a second copy of each chromosome. Now we're ready to go through mitosis. So mitosis, you're going to take all the DNA and split it in half. The other portion is called cytokinesis. That's where we divide everything else. The cytoplasm, all the mitochondria has to get divided up, the endoplasmic reticulum, the Golgi, for example. It all has to get into both new cells. And then you'll have to close off the two new cells with what's called a cleavage furrow. What we're going to see that happens during mitosis is you make what's called the mitotic spindle. See these fibers here? That's how we guide. We send one copy of each chromosome to each new cell. So basically, this is going to be one cell, this is going to be the other. We'll send one copy up, one copy down. Let's go through the stages of mitosis. So mitosis has 4 stages. P-MAT. P-M-A-T. That's the order. So P, the first phase, is prophase. Pros are at the top of their game, they're number 1. Prophase is the first portion of mitosis. So what happens during prophase is you start seeing the DNA condense. Unlike interphase, you can now start to see it. It's winding up on itself, it's getting thicker, it's becoming chromosomes. And then you'll also see the mitotic spindle fiber forming and the centrosomes that make that spindle fiber going to each side. So they're leaving interphase, we're starting prophase. You can start to see those chromosomes condense. And here's a kind of a later prophase. So you can see the chromosomes, but there's no real order to them yet. You can't tell what's happening in there. They're not organized yet. They don't get organized until metaphase. Metaphase is the second portion of PMAT, of mitosis.